My name is Molly Korish, and I will be presenting my project on the citric acid cycle. The citric acid cycle is also known as the Krebs cycle. It's the third step of cellular respiration, and that is a process in which chemical molecules are converted into usable energy. They would enter the body in a form of nutrients such as sugar, so glucose, or lipids, fatty acids. And through the use of cellular respiration, they are converted into a form of energy that can be used throughout the cells of the body. The Krebs cycle step of cellular respiration involves the oxidation of fuel molecules. So again, these would be fatty acids, glucose, or amino acids. Oxidation of these fuel molecules allows the body to transform them into essential coenzymes that can be used in further steps of cellular respiration in order to generate ATP, or energy for the human body. For each glucose molecule that is broken down, the citric acid cycle will occur twice. This is because for each glucose molecule that enters the body, 2 acetyl-CoA molecules are yielded for each of these molecules. Each of these acetyl-CoA molecules will go through the Krebs cycle once each, so twice total for each glucose molecule. The citric acid cycle, or the Krebs cycle, will be occurring in the matrix of the mitochondria of the cell. As the citric acid cycle is the third step of cellular respiration, glucose has previously undergone other reactions, and it starts the citric acid cycle in the form of acetyl-CoA. Each glucose molecule produces 2 molecules of acetyl-CoA, which allows the citric acid cycle to occur twice. The molecule that will combine with acetyl-CoA to begin the citric acid cycle is known as oxaloacetate. For every rotation of the citric acid cycle, 2 carbon dioxide molecules will be produced, as well as 3 NADH molecules, 1 FADH2 molecule, and 1 ATP molecule. While the products of the citric acid cycle are not heavy in ATP or direct energy, the products that can be found from the citric acid cycle will be used in further steps of cellular respiration to generate further ATP for the body. Oxaloacetate will also be regenerated through the citric acid cycle, as it is the molecule that is necessary to kickstart the cycle and will be combined with acetyl-CoA to restart this process. Here is a look at what the citric acid cycle looks like. In gray, you can see the products of each step, and underneath them are the enzymes that will be used to catalyze each step of the reaction. In total, the citric acid cycle has 8 steps, or 8 reactions that combine together to create the full cycle. As you can see at the top, glucose is the starting point, and it is converted to pyruvate, occurring in a previous step of cellular respiration. The product of this previous step, or pyruvate, will be converted into acetyl-CoA and allow for the citric acid cycle to begin. The first step of the citric acid cycle begins with acetyl-CoA reacting with the kickstart molecule, oxaloacetic acid, or oxaloacetate. This reaction will produce citrate, which will be used in the next step. The enzyme that will be catalyzing this reaction is citrate synthase. The second step of the citric acid cycle begins with the citrate that was previously produced in step 1. Citrate is then converted into isocitrate, which is an isomer of the original citrate. This conversion into isocitrate is done through elimination and addition of water, or H2O. The enzyme that will be catalyzing this reaction is cis-aconitate, and again will produce isocitrate. From this isocitrate, we begin step 3. Step 3 involves the enzyme isocitrate dehydrogenase. This will be the enzyme catalyzing the reaction and will occur to produce alpha ketoglutarate. During this reaction, carbon dioxide, or CO2, will be released as a product. As well, NAD+ is transformed into NADH during the reaction. Step 4 of the citric acid cycle begins with the product generated in step 3, which is alpha ketoglutarate. Alpha ketoglutarate will form succinyl-CoA during step 4. Carbon dioxide will be released again during this step, and NAD+ will again be transformed into NADH. The enzyme that will be catalyzing this reaction, or step 4, is alpha ketoglutarate dehydrogenase. This reaction will produce succinyl-CoA. Step 5 will show a phosphate group displacing the CoA group on the previously formed succinyl-CoA molecule. The product created from step 4 was succinyl-CoA, as seen on the previous slide, and will be found with a CoA group on it. During step 5, a phosphate group will displace this CoA and replace it on the molecule. This phosphate will then be passed into the ADP molecule. The addition of another phosphate to an ADP, or adenosine diphosphate, creates adenosine triphosphate, or ATP. ATP is the energy used in the cell, or energy currency used in the human body. So step 5 will generate an ATP molecule that can be used to generate further energy. The enzyme that will catalyze this reaction is succinyl-CoA synthetase. The product of this reaction will be succinate. While in biomolecules, you can expect to see the ADP group bind with another phosphate group to generate ATP. In other molecules, this same similar reaction will occur with GDP. A phosphate will bind to the GDP to create GTP, which again is the next form up when you add a phosphate group. Step 6 of the citric acid cycle begins with the succinate that was formed in step 5. This succinate is then oxidated in step 6 and forms a molecule known as fumarate. During this reaction, FAD will be reduced to FADH2. The enzyme that will be used in this reaction is succinate dehydrogenase. And again, the product will be fumarate. Step 7 of the citric acid cycle involves the addition of water to the previously formed fumarate. Fumarate was the product seen in step 6 and will be the reactant for step 7. Through the addition of H2O, as well as the catalyzing enzyme fumarase, step 7 will yield a product of malate. Step 8 of the citric acid cycle is the final step and will produce oxaloacetate. This is the molecule that was seen at the beginning of the Krebs cycle, or the citric acid cycle, and is the molecule that will be used to react with acetyl-CoA to start the cycle over again. This will begin with the previously formed molecule L-malate and will then be transformed using the enzyme malate dehydrogenase and will produce oxaloacetate. The link to this video can be found at the end of the slides that will be posted on Acorn. Thank you very much for your time and patience.